Cell cycle analysis. If a cell divide, cell go into the different phases. So this phases has a G1, S, G2, M phases. If the cells are not ready to go into the cell cycle, then they are in a quiescent state, so-called G0 state. Also, if the cells go into apoptosis state, the cell size becoming smaller and fragmented, those we call sub-G1 state. During the cell cycle, the DNA contents is changing systematically. When the cells are in G1 state, the DNA amount is 2N, and this is a diploid state. When the cell passes G1 checkpoint, DNA synthesis starts, and then it doubles the amount of DNA. It reaches to 4N state. And during the G2 and M phase, and the cell contains maintain the 4N state of DNA contents. When the cell divide, now one cell becomes a 2 cell, and it goes into G0 or another G1 state. Now it becomes diploid again. If the cells go into the apoptosis and fragmented, then total amount of DNA is less than 2N. This is what we call sub-G1 state. If you track the cell cycle response of a drug compound, especially anti-cancer drug, then by monitoring those effects from cell cycle, we can guess what is the mechanism of this drug A. For the high-throughput single-cell analysis, flow cytometry is a common tool. Here the flow is the flowing the cell through the tube, then one by one those cells can be analyzed. Cytometry's cyto is a cell and metry is a measurement. Through this technology, we can easily measure the contents of DNA per cell, also different properties of the cells. And this flow cytometry technology, easily it can analyze thousands of cells per second. The classical flow cytometry machines utilize a scattering signal to analyze the cells. The most common example is analyzing blood cells. Except red blood cells, the white blood cell, so-called leukocytes, those are immune cells, and they have lymphocytes, monocytes, and granulocytes. Size-wise, lymphocytes is smaller than the other two, and granularity-wise, lymphocyte and monocyte has a low granule inside, but granulocyte, as the name implies, it has a lot of granules inside. The combined cell can pass through the tube, then one by one the cells can flow and emit the irradiated light. So let's say if this cell is interact with the incident blue light, there are two kinds of signal we can detect. One is a forward scattering signal, and usually this signal is proportional to the cell size. So that means if the cell size is bigger, then the forward scattered signal is also big. Another detector on the side detector can measure the side scatter, and this side scattering signal is usually proportional to the granularity. So if the cells have a lot of granule, then the side scatter signal is increased. If you plot those scattering signal, x-axis as a forward scatter, y-axis as a side scatter, we can distinguish different type of blood cells. So, small size of forward scatter and also low granule is a lymphocyte, and bigger but low granule cells, those cells are monocyte. And medium to large cell with high granules, those are defined as a granulocyte. And if the amount of the scattering is low, then usually those are fragmented or debris in the cell mixtures. In addition to the classical scattering signal, fluorescence signal can provide more information. Now, If the cells has almost the same size and physical shape, then how can you distinguish those cells? In this case, if you label those cells with a different antibody with a different color of fluorescence molecules, then we have further tools to classify or distinguish these cells. Two types of cells labeled with a two different color, one green, one orange. Then these mixtures of cells can flow through the tubes. And then when they interact with blue light, now if it has green fluorescence, we can detect the amount of a green signal that can represent the labeled molecules. And also different type of molecule with orange color or green color again. So each cell's relative amount of fluorescence signal can be detected. And based on this information, we can discriminate two types of cells. Using DNA selective fluorescence dye, we can measure the DNA amount per cells. Propidium iodide is a representative of the fluorescence molecule. It has strong fluorescence when it binds to DNA, and the amount of signal, the fluorescence signal, is proportional to the amount of DNA. This is a typical example of cell cycle analysis of actively growing cells. Here, x-axis is the DNA contents, which was measured by PI intensity, and y-axis is the cell number. 200 is similar to 2n state, so that represent G1 or G0 state. 400 is near to 4n, it's a double the amount of DNA, so this represents the G2M. Then the intermediate state is S phase, so DNA contents is increasing up to 400. If the cells go into the apoptotic state, then there is a sub-G1 population can be observed. If it is actively growing and healthy cells, usually we don't see them. Now, usually, even though actively growing cell populations, usually G1 state is the highest population, and then S is ongoing, but a little lower population. G2M, there is a little accumulated state, but once they reaches the G2 checkpoint, then they pass through the M phase. So that population is relatively lower than G1 state. So G1 is the highest population, and G2 and S phase is relatively low populations. If you add doxorubicin to the growing cancer cells, what kind of a change do we expect? Doxorubicin is a topoisomerase 2 inhibitor, and this enzyme is important to DNA synthesis, but not in the middle of a synthesis, it's a later stage. And we analyze those subpopulations. Seems like the S phase and G2M phase is increased. Relatively, G1 is decreased, and the apoptotic population is increased like this. Paclitaxel is microtubule overstabilizer, and this can attack the M phase of the cell cycle. So that's why with the paclitaxel, we can see M phase increased and accumulated, and then G1 and S phase is decreased. This compound is also toxic, so that's why this apoptotic population is also increased. So this kind of M phase accumulation we call M phase arrest of the cell cycle. How about the cisplatin? Cisplatin is a DNA alkylating agent, and it binds to the DNA, and that interferes with the DNA synthesis. So if you analyze those cell cycle change, S phase is increased, and the phases G1 and G2 is decreased. And also there is a toxic cells, apoptotic cell population we can observe. So with the cisplatin, we can expect S phase arrest. So that means S phase population in the middle of S phase population is increased. It's a dangerous state, and eventually it cannot survive and it can die.